new cells are formed from the division of pre-existing cells. In other words, every cell is derived from another cell. In unicellular organisms, cell division is the means of reproduction, just as we see amoeba divides into two. Some changes take place in a cell before division to prepare it for dividing. Once the cell is ready, it divides. In multicellular organisms, new individuals developed from a single cell, the zygote, by cell division. Cell division also forms the basis of replacement of old and worn-out cells. So, we see that cell division maintains continuity of living matter generation after generation. A special form of cell division is involved in the production of the sex cells, which contain half the number of chromosomes as that of parent cell. This division is known as meiosis. There are several features unique to meiosis. Most importantly, it produces genetic variations. This happens because of pairing and genetic recombination between homologous chromosomes. Simple example of this variation can be seen in this family photograph. Although there is a resemblance among different family members, but none of them resemble exactly. Observe again.
ਕੀ ਤੁਸੀਂ ਕਦੇ ਸੋਚਿਆ ਹੈ ਕਿ ਤੁਸੀਂ ਬਿਲਕੁਲ ਆਪਣੇ ਪਿਤਾ ਦੇ ਵਾਂਗ ਕਿਉਂ ਨਹੀਂ ਦਿਸਦੇ ਅਤੇ ਤੁਹਾਡੇ ਪਿਤਾ ਜੀ ਵੀ ਬਿਲਕੁਲ ਤੁਹਾਡੇ ਦਾਦਾ ਜੀ ਵਾਂਗ ਕਿਉਂ ਨਹੀਂ ਲੱਗਦੇ? ਅਜਿਹਾ ਕਿਉਂ ਹੁੰਦਾ ਹੈ? ਇਹ ਸੈਲ ਡਿਵੀਜ਼ਨ ਦੌਰਾਨ ਜੈਨੇਟਿਕ ਮਟੀਰੀਅਲ ਵਿੱਚ ਹੋਣ ਵਾਲੀ ਵੇਰੀਏਸ਼ਨਸ ਦੇ ਕਾਰਨ ਹੁੰਦਾ ਹੈ। ਅਸੀਂ ਅੱਜ ਇਸ ਖਾਸ ਸੈਲ ਡਿਵੀਜ਼ਨ meiosis ਬਾਰੇ ਪੜਾਂਗੇ। Meiosis or reductional division is a division which forms gametes that are essential for sexual reproduction. Meiosis is a double division which occurs in a diploid cell and gives rise to 4 haploid cells, each having half the number of chromosomes as compared to the parent cell. It is during fertilization that 2 haploid gametes fuse to form a diploid zygote, which by mitosis forms a diploid adult. So as you know, in our today's class, we will explore the process of meiosis. But first, let me share the learning objectives for today's class.
On completion of this topic, learners will be able to define meiosis, explain the importance of meiosis in sexual reproduction, describe each step of meiosis 1 and meiosis 2. Draw diagrams depicting meiosis 1 and meiosis 2.
Meiosis एक तरह ਦੀ ਸੈਲ ਡਿਵੀਜ਼ਨ ਹੈ ਜੋ ਕੇਵਲ ਸੈਕਸ਼ੂਅਲੀ ਰੀਪਰੋਡਿਊਸਿੰਗ ਆਰਗੇਨਿਜ਼ਮਸ ਵਿੱਚ ਹੀ ਹੁੰਦੀ ਹੈ। ਇਸ ਰਾਹੀਂ ਹੈਪਲੋਇਡ ਗਮੀਟਸ ਪ੍ਰੋਡਿਊਸ ਹੁੰਦੇ ਹਨ ਜੋ ਅੱਗੋਂ ਹੋਰ ਡਿਵਾਈਡ ਨਹੀਂ ਹੁੰਦੇ ਬਲਕਿ ਫਿਊਜ਼ ਹੋ ਕੇ diploid zygote produce ਕਰਦੇ ਹਨ। Do you know where does meiosis occur? Meiosis occurs in special cell types and at a specific period. Meiosis occurs in diploid germ cells of sex organs in testes to form the male gametes called spermatozoa and in ovaries to form female gametes called ova. Let us not get into more intricacies about meiosis before knowing how exactly it takes place.
As I previously discussed, that this type of cell division occurs only in special type of cells. These special types of cells are diploid germ cells of sex organs of male and female. During the growth of a male, undifferentiated germ cells start taking the shape of sperm cells. When a male reaches puberty, these diploid germ cells present in seminiferous tubules of testes undergo meiosis to produce male sex cells known as spermatozoa. Similarly, in a female, germ cells present in ovaries divide and complete meiotic division to form ova. These spermatozoa and ova, thus produced, are haploid cells. They fuse during fertilization to form a diploid zygote. In this way, the total number of required chromosomes is restored.
ਹੁਣ ਅਸੀਂ ਇਹ ਜਾਣ ਗਏ ਹਾਂ ਕਿ ਗਮੀਟਸ ਇੱਕ ਸੈਕਸ਼ੁਅਲੀ ਰੀਪਰੋਡਿਊਸਿੰਗ ਆਰਗੇਨਿਜ਼ਮ ਦੇ ਲਾਈਫ ਸਾਈਕਲ ਵਿੱਚ ਹੈਪਲੋਇਡ ਫੇਜ਼ ਹੈ। ਫਰਟੀਲਾਈਜ਼ੇਸ਼ਨ ਜੋ ਕਿ ਮੇਲ ਅਤੇ ਫੀਮੇਲ ਗਮੀਟਸ ਦੀ ਫਿਊਜ਼ਨ ਦਾ ਰਿਜ਼ਲਟ ਹੈ। ਇਸ ਦੇ ਨਾਲ ਆਰਗੇਨਿਜ਼ਮ ਦੀ ਲਾਈਫ ਦਾ ਡਿਪਲੋਇਡ ਫੇਜ਼ ਰੀਸਟੋਰ ਹੋ ਜਾਂਦਾ ਹੈ। Let us learn about the mechanism of meiosis. Meiosis functions to reduce the number of chromosomes to half. Each daughter cell that is produced will have 1/2 as many chromosomes as the parent cell. Meiosis comprises 2 successive nuclear divisions with only one round of DNA replication. 4 stages are present in each nuclear division. So we can say that meiosis involves 2 sequential nuclear divisions, meiosis 1 and meiosis 2. Each meiotic division consists of the following phases. Meiosis 1, a reductional division, is divided into 4 subphases: Prophase 1, Metaphase 1, Anaphase 1 and Telophase 1. Similarly, meiosis 2 is also divided into subphases which are like this: Prophase 2, Metaphase 2, Anaphase 2 and Telophase 2. Let us discuss meiosis 1. Meiosis 1 is initiated once the parental chromosomes replicate to form identical sister chromatids. Let us see each of its stages one by one with the details of all changes taking place at each stage. The first stage is Prophase 1.
Prophase 1 of the first meiotic division is typically longer than other stages. Prophase 1 of the first meiotic division is a more elaborate process than prophase of mitosis. During prophase, homologous chromosomes come together and lie in pairs to form bivalents or tetrads. Prophase 1 has been further divided into 5 subphases based on changes in behavior of chromosomes. 5 subphases of Prophase 1: leptotene, zygotene, pachytene, diplotene, and diakinesis. Leptotene of Prophase 1. This stage is marked by the first appearance of the chromosomes when the chromosomes are in their most extended form. They look like strings having beads on them. The beads are known as chromomeres. The nucleus enlarges and the chromatin fibers shorten and elongated chromosomes start becoming clear. Although the chromosomes are replicated, but the chromatids are not yet distinguishable. one of the centriole pair begins to move to the opposite side. Both the centriole pairs or centrosomes develop asteral rays, thus forming an aster.
In the cell undergoing meiosis, there are two sets of chromosomes. That is, the chromosome number is diploid. There are two similar chromosomes of each pair. These are called homologous chromosomes. One of the chromosome of the homologous pair comes from the male parent, that is, paternal homologue, while the other is passed on from the mother, which is maternal homologue. The homologous chromosomes resemble each other in the position of their centromeres, shape and size. This we will see in next stage of prophase 1. During this stage, the homologous chromosomes are seen as paired units. The chromosomes become shorter and thicker, and in some cells they remain attached to the nuclear envelope at the points near the aster. Let us see what happens during zygotine.
Zygotine of prophase 1. Pairing of homologous chromosomes takes place. The two chromosomes of each homologous pair get attached to each other laterally or come to lie side by side. Pairing is such that the genes of the same character on the two chromosomes come to lie exactly opposite. This pairing of homologous chromosomes is known as synapses. On account of synapses, chromosomes form pairs which are known as bivalents. The number of bivalent is half the number of total chromosomes. Depending upon the place of origin of pairing, synapses may be procentric, that is, starting from centromere and proceed towards the end. Proterminal when pairing starts from end and proceeds towards the centromere. Intermediate when pairing starts anywhere between centromere and the end. We see that a pair of homologous chromosomes consists of two chromosomes. That is, a bivalent can also be called a tetrad as it has 4 chromatids. The two chromatids belonging to the same chromosome are called sister chromatids, whereas chromatids belonging to different chromosomes of a homologous pair are termed as non-sister chromatids. Next stage is pachytene. Pachytene of prophase 1. Due to further condensation of chromosomes, the tetrads are now clearly visible. Sometimes, exchange of genes or crossing over between non-sister chromatids of homologous chromosomes occurs at the points called recombination nodules. Crossing over is a process of exchange of genetic material or chromatid segments between two homologous chromosomes. Crossing over is an enzymatically controlled process and is regulated by recombinase enzyme. Crossing over leads to recombination of genetic material on the two chromosomes. Next stage is diplotin of prophase 1. Homologous chromosomes tend to separate from each other except at the sites of crossover. Usually, it is recognized by the dissolution of synaptonemal complex means the synaptonemal complex begins to break down. The chromatids begin to pull apart, revealing this X-shaped structure known as chiasmata. At first, the chiasmata are located at the sites of the recombination nodules, but later they migrate towards the ends of the chromatids, which is called terminalization. Diakinesis of prophase 1. This is the last stage of meiotic prophase 1. This is marked by the end of the terminalization of chiasmata. The chromosomes get more condensed. This is followed by the chromosomes recondensing in preparation for metaphase. Nucleolus thus disappears and nuclear envelope also dissolves. Spindle fibers start appearing. This stage represents transition to metaphase. Let us see what happens during next stage, that is metaphase 1. Metaphase 1. During metaphase 1, bivalence, that is each unit composed of 2 chromosomes, or we may say 4 chromatids, align themselves at the metaphase plate. Metaphase 1 is characterized by a fully formed spindle. By the end of this stage, bivalents independently attach themselves to metaphase plate. The orientation is random. The maternal homologue of each bivalent may be oriented towards either pole, and similarly, the paternal homologue of each bivalent may also be aligned towards each pole. This means that all possible combinations of chromosomes can occur in the daughter cells. Anaphase 1. Now the process moves to next stage, Anaphase 1. Chromatids forming chiasmata separates. As you see, the homologues of each bivalent separate and moves to opposite poles. Here we notice that each chromosome still has 2 chromatids. Sister chromatids remain associated at their centromeres. Telophase 1. This is the last stage of meiosis 1. Here, nuclear envelope and nucleolus reappear. Cytokinesis follows and a dyad of cells is formed.
So this was the last stage, telophase 1 of meiosis 1. Some species do not have a proper telophase 1. That is because the next division comes up so quickly that there is no need to bother with all that work, rebuilding the nuclear membrane only to soon break it up again is a waste of energy, because that work would just be reversed in the next prophase. Now next division of meiosis has to begin. There is always a stage between 2 divisions, and this stage is known as interkinesis. This stage is very short-lived. This stage is similar to interphase between 2 mitotic divisions, except that DNA replication does not occur here. Why? Chromosomes in each haploid cell are in replicated form with 2 chromatids attached at a centromere. The two chromatids are no longer sister chromatids, since crossing over has made them unique hybrids. Let us see how exactly these chromosomes behave in the second part of meiosis.
Meiosis 2. Meiosis 2 begins without further replication of the chromosomes. Both the cells formed during meiosis 1 start dividing, and the first stage is as usual prophase. Here, this stage is known as Prophase 2. This is Prophase 2, the first stage of meiosis 2. The Prophase 2 begins with dissolution of nuclear envelope and nucleolus. Aster and spindle formation start taking place from the centrioles. Chromatin material condenses into chromosomes. Next stage is metaphase 2. The chromosomes align with their centromeres on the equatorial plane on the spindle fibers. The kinetochores of the sister chromatids point toward opposite poles. Each centromere gets joined by two chromosomal fibers. Anaphase 2. Anaphase 2 begins with the simultaneous splitting of the centromere of each chromosome. By this splitting, sister chromatids move towards opposite poles of the cell. Next stage is telophase 2. Here, nuclear envelope is formed again, nucleoli reappear, and chromosomes decondense. Astral rays and spindle fibers disappear. The last meiotic division effectively ends when the chromatids arrive at the poles. Cytokinesis. The cytokinesis stage is always present and occurs by cell furrow formation in animal cells and by cell plate formation in plant cells. Each daughter cell now has half the number of chromosomes. Cytokinesis produces 4 haploid daughter cells.
ਸਾਈਟੋਕਾਈਨੇਸਿਸ ਦੇ ਅੰਤ ਵਿੱਚ ਅਸੀਂ ਵੇਖਿਆ ਕਿ ਇੱਕ ਡਿਪਲੋਇਡ ਸੈੱਲ ਤੋਂ 4 ਹੈਪਲੋਇਡ ਸੈਲਸ ਪ੍ਰੋਡਿਊਸ ਹੁੰਦੇ ਹਨ. The main aim of meiosis 2 is to separate genetically modified chromatids of each homologous chromosomes formed during crossing over in meiosis 1. Due to crossing over of chromatids, each gamete can contain chromosomes with different types of genes. Thus, meiosis produces new combination of genes due to crossing over, which results in genetic variability. Without meiosis, chromosome numbers would continually increase. Meiosis ensures that daughter cells receive one of each kind of chromosome, precisely halves the parental chromosome number. Meiosis and fertilization both work in coordination to maintain fixed number of chromosomes generation after generation. It is so because sexual reproduction without meiosis will result in tetraploidy, means 4 times more chromosomes. If meiosis 2 fails after completion of meiosis 1, the stage is known as brachymeiosis. Without meiosis, chromosome numbers would increase continuously. So this was all about meiosis. Let us now summarize what we have learned so far. Meiosis, a process involving 2 consecutive cell divisions, is an essential component of sexual reproduction. It produces haploid gametes from diploid parental cells, with each gamete containing a unique mixture of the parental genes. Each of the two cell divisions has phases that superficially resemble the phases of mitosis, but actually you have seen quite different events occur during meiosis. Let us have a glimpse of these divisions once again. Interphase. Before meiosis 1 begins, chromosome replication occurs. That means genetic material is duplicated. First division of meiosis, meiosis 1. Prophase 1. Prophase 1 is a complex and prolonged phase consisting of 5 subphases: leptotene, zygotene, pachytene, diplotene, and diakinesis. During prophase 1, homologous chromosomes pair and exchange of genetic material takes place as a result of crossing over. Metaphase 1. Homologous chromosomes align at the equatorial plate. Anaphase 1. Homologous pairs separate with sister chromatids remaining together. Telophase 1. Two daughter cells are formed with each daughter cell containing only one chromosome of the homologous pair. Second division of meiosis, meiosis 2, gamete formation. Prophase 2. DNA does not replicate and the nuclear membrane and nucleoli disappear. Metaphase 2. Chromosomes align at the equatorial plate. Anaphase 2. Centromeres divide and sister chromatids start migrating towards pole. Telophase 2. Cell division is complete. 4 haploid daughter cells are obtained. One parent cell produces 4 daughter cells. Daughter cells have half the number of chromosomes found in the original parent cell and with crossing over are genetically different. Meiosis differs from mitosis primarily because there are 2 cell divisions in meiosis, resulting in cells with a haploid number of chromosomes. So now we come to the end of today's topic. Here is a quick test for you to find out how much you have actually absorbed. The first question is, what is meiosis? The answer is, meiosis is a double division which occurs in a diploid cell and gives rise to 4 haploid cells, each having half the number of chromosomes as compared to the parent cell. The next question is, chromosomes that are similar in size and shape and contain genes for the same traits are called homologous chromosomes or dash. The answer is, chromosomes that are similar in size and shape and contain genes for the same traits are called homologous chromosomes or homologues. My next question is, at which phase of division does the nuclear membrane disappears? And the answer is prophase. The next question is, the transition phase between meiosis 1 and meiosis 2 is called dash. The answer is, the transition phase between meiosis 1 and meiosis 2 is called interkinesis. The next question is, at which stage chromosomes come to lie over equatorial plate? The answer is metaphase. Here comes the next question. At which phase genetic recombination during meiosis takes place? The answer is pachytene. Next question. During which phase of meiosis do tetrads form? Anaphase 1, prophase 2, prophase 1 or Anaphase 2? And the answer is tetrads are formed during Prophase 1. The next question is, what are sister chromatids of chromosome? The answer is, the two chromatids belonging to the same chromosome are called sister chromatids of chromosome. Next question is, which enzyme regulates crossing over? The answer is, recombinase enzyme regulates crossing over. The next question is, fill in the blank. Meiosis 1 produces 2 dash cells, each with dash chromosome, whereas Meiosis 2 produces 4 dash cells, each with dash chromosome. And the answer is, Meiosis 1 produces 2 haploid cells, each with double chromatids chromosomes, whereas Meiosis 2 produces 4 haploid cells, each with single chromatid chromosomes. The next question is, if you have to count the chromosomes in the cells of onion root tip, which stage will you look into? The answer is metaphase stage. Next question. At which stage of division bivalents are formed? The answer is zygotine. Next question is, if Meiosis 2 fails after completion of Meiosis 1, the stage is known as? Answer, brachymeiosis. Next question. If at the end of meiosis the 4 daughter cells have 4 chromosomes, how many chromosomes were in the mother cell? The options are 2, 4, 8 or 16. And the answer is 8. The mother cell had 8 chromosomes. ਮੈਨੂੰ ਉਮੀਦ ਹੈ ਕਿ ਤੁਸੀਂ ਅੱਜ ਦੇ ਟੌਪਿਕ ਨੂੰ ਇੰਜੋਏ ਕੀਤਾ ਹੋਵੇਗਾ। ਧਿਆਨ ਦੇਣ ਲਈ ਬਹੁਤ-ਬਹੁਤ ਧੰਨਵਾਦ। ਅਗਲੀ ਕਲਾਸ ਵਿੱਚ ਨਵੇਂ ਟੌਪਿਕ ਨਾਲ ਫਿਰ ਮਿਲਾਂਗੇ। Till then, goodbye.